Hi class, we are back in the lab today and I'm going to give you a brief explanation of how to make a streak plate. Now, there really is no lecture that goes with this because the streak plate is a technique that we learn how to do, and then you can do a streak plate on any kind of media that's in a plate. So we do a streak plate whenever we have a sample of bacteria and we want to get what is called an isolated colony. So if you remember that PCA plate from the very first video of microorganisms in the environment, where we had several different kinds of colonies and several different kinds of bacteria on one plate, that's what we want to not do on the streak plate. So basically we would take one of those colonies and then do a streak plate with it. That way we could get a pure sample. Another way to do that is with a broth culture. So I'm going to use a broth culture today to demonstrate the streak plate for you. Now these are my cultures, so these are already pure. I know that there's no other kind of bacteria in them except for what's written on the tube. And so this is a way that I keep my bacteria fresh, as I streak them on plates every couple weeks. So what you want to do whenever you get a streak plate, and this is just TSA or tryptic soy agar, and it's a general purpose media that all of my bacteria can grow on. And so you want to label your petri dish. So I've got it labeled what kind of bacteria this is going to be, and then I have a T written on it. Now, I don't write T's on all of mine, but I just wanted to do this because this is how the diagram is done in your book. And so basically what you're going to do is we're going to put the bacteria in this first quadrant, okay, using a loop. And then we're going to flame our loop, and then we're going to drag over a few cells into this second quadrant, flame our loop again, and then drag over the last bit of cells into this third quadrant. The whole point of this is to get an isolated colony. So we're going to put lots of cells on the plate here, then we're going to dilute them down, to where hopefully over here in the third quadrant, we only have a few cells, and then those will give rise to our isolated colonies, where it's one colony by itself. And then we use that colony to then do stains with and any other tests with, because that colony came from the same bacteria. So we know that it's all the same strain. And so that's what we're going to do with the streak plate. So I'm going to, of course, we're going to want to use our Bunsen burner whenever we're doing this, because we want to use Aseptic Technique. And I actually have 2 loops that I'm going to use. This just makes the process quicker because you don't have to wait for them to cool. So I'm going to flame my loops, and I'm going to put them down to the side and let them cool. And remember, we're practicing Aseptic Technique, so you don't want to touch the loop to anything. That's why I have it on the edge of the table. You don't want to touch anything while it's cooling, okay? And also, you want to give your bacteria just a little mix before you streak them, just so you can get, and because sometimes the cells like to settle at the bottom of the tubes, and we want to make sure that we've got it all mixed. I'm going to do all 8 of these, but I'm just going to show you the demonstration with one of them, okay? So my loop is cool. So always want to keep your plate inverted while you're doing this, because you want to prevent airflow. We want to prevent contamination. So my first bacteria is going to be E. coli, so I'm just going to dip in once. Then I'm going to streak in the first quadrant back and forth. Now, then I'm going to flame my loop. Now my other loop is already cool, so I'm going to drag over twice to pick up some cells, then stay in quadrant 2, flame my loop. And remember, we're not stabbing into the agar, we're gliding on top. And then start in quadrant 2, then stay in quadrant 3. Now, if you stab a little bit like I did there because I was at a weird angle, it's okay. The bacteria will be fine, but you just don't want to do that a whole bunch. And just making sure you flame your loop after. And that's how you make a streak plate.